Hello, everybody. What are you doing?
Hi, Irvin.
Hi. So you had some technical issues?
Yes.
Ah, life is hard sometimes.
But we are ready. Stephanie told me that both Lilith and Cass would like to present. Who volunteered to go first?
Who was
Volunteer.
okay.
I can go.
Okay, great. Did you share the link in the chat box?
Yes, it's the second. I mean, Stephanie.
Steph, that one. Great.
The second link, yes.
Okay, see if you can share the screen. Great.
Can you see it?
Yes, it's big. I can read it. Perfect.
Okay, cool.
Great.
So hello everyone. I want to show you this paper. The name is Redefining Varicose Projection Astrocytes in Primates. So in short, this paper is about a cell that is present just in humans and apes. This cell looks like on a scheme like this. So it's basically a typical astrocyte with just a couple of long processes. And on these processes, you can see nodes. And these nodes are called varicosities. So that's why the name of the cell is varicose projection astrocyte. And you can see this cell also on these amazing pictures. So there is, for example, soma of the cell and the long process with its varicosities. And for some reason, according to previous study, these cells are present just in a human brain and in chimpanzee. But authors are saying that these evidence are based just, first of all, only on pathological human brain and on just a chimpanzee, one chimpanzee. So we actually don't know if this cell is specific just on, if this cell is specific actually just for primate brain, just for chimpanzee, or we can also find these cells in other primate brains, for example, in more primitive primates. So that's why these authors decided to examine huge range of different primates and different great apes and select only human with no defined neurological disorder. So this is the range that they had. It's quite impressive in my opinion, and what did they found is that only these cells, as these astrocytes with varicosities, were present only in 5 species, types of species, yes, and including humans and 4 apes. So none of other primitive primates, they didn't have these cells at all. And what was surprising is that in contrast with previous study, they didn't find these cells in chimpanzee. So they had 6 cases of chimpanzee and none of them included this type of cell for some reason. And yeah, so they thought, okay, maybe it is connected with size of the brain, maybe big animals, they have these astrocytes with varicosities, but that was not the case because they also examined brains of African elephant and giraffe and whale, so none of them had these astrocytes. And also interesting is that actually not all human brains had these astrocytes. So half of the sample had these astrocytes. And also if we look at gorilla, there are only 3 out of 5 brains included these cells. So this makes authors think that actually it is not a distinct astrocyte, like a special type, but it's an already known astrocyte with some deformation, which is caused by some condition that they don't know for now. So, and most probable it is like fibrous astrocyte because it has it is located in the same areas, so it's like the fourth layer and in white matter, and it has more the same length and size of the processes. So that's why they are thinking probably it's a normal fibrous astrocyte, but with some deformations caused by unknown condition. So, and then they thought that, okay, maybe it's connected with some inflammatory processes, but that was not the case because they measured the density of microglia, and it didn't have any correlation with the presence of these cells. So it is not connected with inflammatory processes. Yeah, so what did they also found is, so they also go more into detail with the human brain. So I forgot to mention that all these species, they analyzed just frontal cortex, except human. They, in addition to studying frontal cortex, they analyzed other cortical areas, and they found that these people who had in their brain this cell, they had in frontal cortex, they had these cells also in other parts of brain. Whereas none of these people, from these 7 people, they didn't have these cells, these formations on astrocytes, neither in frontal cortex nor in other areas. They didn't examine the other primates and apes if it is present in other cortical areas. It was just frontal cortex for other species. Yeah, so this is yeah, also interesting was that in contrast with previous study, they said that the morphology of the cells of these cells are similar in human and apes. So in the previous study, they other authors, they claimed that this is just it is more complex in human, the morphology of varicose projection astrocytes. But in this case, they they measured different aspects of this cell, like the length of processes, the shape of the soma, and everything was similar. So there was no statistical difference between these cells. And yeah, so I guess yeah, so overall, I can say about this paper that it is a nice paper that provides us some observation about what is happening with inside the brain of huge variety of different species, and it kind of gives us some insights of the reason why we will have this formation of astrocyte on the processes of the astrocyte, but unfortunately it didn't give us some solid theory why we have these formations on some processes of some species and not in all cases. So the only hypothesis that they tested is about inflammatory processes, and they also tried to say that maybe it is connected with the age of participants, of cases, and it was also not the case. They didn't find any correlation, although the youngest human participant, was didn't have these cells as well. Yeah, and then they also think, okay, maybe it's connected with some stress or other condition, but they couldn't find. They cannot provide this, some evidence for this hypothesis. So for now, it's kind of open question, and maybe you have some ideas why we can see these varicosities on some astrocytes and only in great apes and in humans. Yeah, and also another thing is just it appeared that there is interlaminar astrocyte, another type of astrocyte, and for some reason, if a brain had these various varicose projection astrocyte, then intralaminar astrocyte also had this formation on its processes. And so they thought maybe it's connected with the tissue processing, that they damaged something, I don't know. But they said that it's also not the case, because other astrocytes, like, for example, as other astrocytes, for example, protoplasmic astrocyte, they didn't have any deformation. So it's just specific connected with interlaminar and varicose projection astrocyte. But the main line, again, it's just they're thinking that it is fibrous astrocytes with some deformation caused by, for now, unknown condition. And this deformation can be found only in human and some great apes. Yeah, basically that's it. I feel like it's interesting to know the reason why we can observe these varicosities. Maybe I thought, I don't know, maybe somehow it may be connected with, could be connected with Alzheimer's disease. Maybe it's some type of problem that was not that they haven't noticed. Maybe it's like very early stages of Alzheimer's disease that cause this deformation, or is a cure with this disease. I don't know, but these patients didn't have known neurological disorder. These are human cases, and cause of death are not connected with neurology. But this could be maybe some initial stages, and it just hasn't been yet determined. Yeah, so I think it was super fast, I don't know, but this is basically what caught my attention, and I will be happy to discuss this with you, and maybe you have some ideas why this could happen in different brains.
Very cool paper. Thank you, Lilith. I can see Leah already has a question. Fire away.
Yes, it was connected on the last things that you were discussing, because so I was really curious which is the function of these varicosities. And in the discussion, it seems that it's something that is pathological. Instead, when you were presented, I thought that was something like in an evolutionary perspective, so all in the frontal lobe, something that could had a functionality or something. I didn't see that where you were presented, because I didn't know. I never heard about it, and but no, it seems that it's more in a pathological way.
They are not saying pathological. They are using the word like under some conditions, so we don't know.
Okay.
What it is, some like microenvironment of the brain, and we don't know the lead, but the cause. But yeah, there are. They didn't say that it is something cool. They said it's maybe connected with aging, with stress. So it's not about pathology. It's more about some negative conditions more. But we don't know. Maybe they didn't provide evidence that it is connected for sure with stress or not. So with aging, they didn't find any correlation again. It's not connected with inflammation. Yeah.
But if I get it well, in the humans is more present related to the other species or not?
It's present indeed present in frontal cortex of 4 types of species, not chimpanzee, except from chimpanzee and siamang. In case of humans, they also found these cells in other cortical areas, but it doesn't say that these cells are not present in other cortical areas of apes. They just didn't look at it.
Understood.
Yes. So no, but the thing that also, like in other paper where I tried to find, maybe there is a link with this astrocytes and Alzheimer's disease. And in that paper, when they are discussed, and it's a new paper, also 2021, but just published in March. And they are talking about varicose projection astrocyte, like it is only present in primates. So this result is kind of new, I guess, so that also present in human. And it's not that maybe popular opinion now. I mean, it's maybe new, it's just they are just trying to figure out, and with this amazing method, and there are cool pictures like this.
Yeah. No, it's amazing how you can really go in depth.
Yes. Yeah, and you can find them why they are thinking it is some condition of fibrous astrocytes, because fibrous astrocytes can be found also in white matter, whereas the other one is present only in the cortical area. So in this case, for example, in orangutan, you can find them both in white matter and in fourth and fifth layer of cortex.
Yeah, thanks. Amazing.
Are there any other questions? No? Everyone happy with it? Great. I missed the beginning of the paper. I'm very sorry about it. But I thought it was super interesting. And also getting so many species for comparison, not that easy. So I think it is quite interesting. And you did a good job in highlighting what's new about it and why this is actually an exciting paper. So you'll have the link in the chat. I'm going to also put the link on our YouTube channel for people to read the full paper. And if there is no further questions, we can move on to our second presentation today from Katrin.
Hello, nice to meet you, everyone. I'm Katrin. I'm joining you for the session, and I will share my screen now. There we are. Can we all see? Yes, okay. All right, so the paper I chose today is from a group from Boston University and Madrid University, and it's about the cortical spectrum, and essentially the authors compare histological methods with histological measurements with MRI measurements. What really interested in me in this paper is, well, I've always been really interested in histology and cytoarchitecture, but it's always seemed very inaccessible because it seems like you need to be very highly trained in order to study it. And you always can have a smaller sample size and it's very labor intensive. And the fact that they are actually comparing it with something a little bit more accessible like MRI data, I found very fascinating. And so the main idea in this paper is this idea of the cortical spectrum, which is essentially the idea that the cortical surface, the structure of the cortical surface varies in like a gradient and structured manner from limbic areas onto association cortices and finally sensory areas. I guess the authors kind of start with saying that this idea is kind of like a medium between two extreme ideas as to cortical organization. So on the one hand, we can look at the cortex as cortical areas with very sharp borders, and we can think of examples such as Brodmann maps, where cortical areas are defined with very sharp borders, and in fact, numbers of these areas are very arbitrary, and they don't really tell us anything about how these areas are connected. Or on the other hand, we can look at the cortex as completely homogeneous. The areas are not really different from each other, and the only way that we can tell them apart is based on their extrinsic connections. But we know that those are two very extreme ideas, and that's not actually what the evidence shows us. And so they, what the evidence shows us, and so essentially what they do is they have a look at two different measurements. They look at some measurements which are taken from site architecture, such as myelin staining and staining for pyramidal cells with the specific protein called SMI33. And they also look at myelin structure, which has been taken from measurement of MRI, and which is taken from the ratio of T1 and T2 weighted imaging. So they essentially what they say about this cortical gradient, which spans across the whole cortex, is because we can think about it as kind of like a continuous gradient, but they suggest this idea of the cortical types, which, if I understand correctly, is essentially the idea that you can discretize this gradient into smaller units of regions, which have similarity based on laminar characteristics, such as number of cortical layers, prominence of these layers, or sharpness of boundaries. And all of these structures covary together between the regions. So it's a bit kind of easier to group those regions together. And unlike something like Brodmann's map, those characteristics are not arbitrary. They're based on what we actually see in the cortex. And we can have a variety of different types. So we can have from up to 3 to up to 8. But we can still compare just because the basis of the gradient is just it's still basically comparable no matter how you divide it, essentially. Okay, so what they did is they took for the staining analysis, they essentially took these. So they only looked at the rhesus macaque monkey. And they had slices from the left and the right hemisphere, and each hemisphere they stained for different things. For example, the left hemisphere they stained for myelin, as you can see on the left of this figure, and for the right they stained for the SMI32 staining, which is for the pyramidal cells. And what they did, so basically the differences. So I guess we can see in the difference of the size of the slices. I think they explain comes from the constraints of the staining method. So I think they kind of shrink when they use this protein staining, whereas with myelin, they grow in as we see them on the left. Sorry, let's have a look at my notes. Right. So what they did, they were really interested in the area that starts from the pure matter up until the superficial white matter. So they only really looked at the gray matter. Now, as for so I guess one of the problems that they point out with staining methods is that it is very labor intensive, and you can't really compare a computational method compared to a specialist actually looking at it and working like very meticulously on a stain. But they also mentioned there is a little bit of possibility for subjectivity bias. And so they say it is kind of necessary to at least have like a granular computational approach that allows us to look at multiple areas at the same time. And so what they do is they essentially develop this computational approach. The first things they do is they have actual researchers have a look at the original image of the staining and have a look at the binarized image of the staining. So what they do is, so what the researchers have to do is they essentially have to set a threshold for this binarization in such a way that it balances both the noise in the image with the actual laminar structures that we can see. And they did it with several different researchers. And they actually found out that they were all in agreement in the way the threshold was set. It was all completely blind as well. And so what they did with the sort of binarized images that they had is that they subdivided them into different sections based on structural connectivity. And for each section, they calculated the relative gray matter level by just averaging. And what they actually found was they were able to subdivide one photomicrograph into up to 25 different little areas, which is quite a lot compared to what we traditionally think about cortical layers. So I usually think about layer 1 to 6, but they even managed to find differences for up to 25. And they did that for both their myelin staining, both their sort of pyramidal cell staining. And what I found quite exciting about the results is that, what they did, firstly, what they did is that they divided all their regions, all their 78 cortical regions that they were interested in testing into these cortical types. So the cortical types that they've chosen start from agranular, which characterized by having kind of a weak layer 4. And those were areas such as limbic cortices, agranular and dysgranular, moving on to eulaminate 1 and 2, which have slightly better, slightly clearer layer 4 and a little bit more differentiation between the other layers. That's such as temporal cortex, parietal and frontal areas. And then finally, eulaminate 3 and conio cortex, which have the clearest lamination in all the cortical types. So that would be areas such as V1, areas in S1. And based on this differentiation, they applied it onto their data, and they found that they could see this kind of clear organizational gradient, which starts from the most laminated to the least laminated, which kind of corresponds with this idea that they had initially about the cortical spectrum. And they were able to find that for both myelinate staining and both for their pyramidal cell staining. But then most excitingly, they did the same thing with the T1, T2 myelin data. They essentially plotted the T1 versus T2, and they found kind of a similar gradient of organization. Now, I guess the thing which you will notice is that the gradients go in a slightly different directions. For example, if we look at here, it starts from coniocortex to agranular, whereas in MRI, it's kind of the where did my image go? It's kind of in the opposite direction. And they say this is because the model that they apply to generate in order to generate their data, essentially the sign of the direction of the gradient is kind of arbitrary. Well, not arbitrary, but they're able to reverse the sign without actually affecting the data. I think it just has to do with complicated maths that unfortunately I can't explain. But yeah, overall, they found very strong agreement between all of their measurements, which I found really interesting, that you can see that specificity not only at histological staining level, but also at MRI level, and we can actually compare. Later on, they did kind of the same thing by measuring kind of a z-scored version of all their measurements for all of their different cortical types. And they found, again, similar patterns of this sort of differentiation between all of their regions. And so to me, that looks like their methods actually looks very good. It looks like it's really doing what it should do. And I don't know, I find it really fascinating. But yeah, I guess what it actually shows us is that this cortical spectrum does actually exist. We can see it on different levels, starting from histology to MRI staining. Yeah, I feel like it makes that kind of analysis a little bit more accessible to other researchers as well. Yeah, and yeah, I really enjoyed this paper overall. I have to say, I struggled a little bit with the model that they used. I think essentially they took all of their analysis and generated this massive data set, and then they had to apply a dimensionality reduction method, and that's what actually allowed them to have a look at the data in kind of a more comparable way. But yeah, I'm really excited for this. I thought it was a great, cool paper. I don't know if you guys agree, but yeah.
Cool. Thank you so much. It's amazing to see how excited you are about this publication. And indeed, we had a little chat on the side trying to work out the embedding and dimension reduction methods as well. Thank you. Any questions? You guys are very quiet today. That's new. Leah.
I can go. I have questions. Thank you. It was super interesting, and I really love the images. Yeah, I was trying to scroll down in the paper to see like which method did they used, but I couldn't find it for the embedding. And but yeah, my curiosity was so this cortical spectrum is well, if we go like from the inner layer to the outer layer, like is just in that direction, or there are variation like across? Because otherwise, to me, maybe it's just a fanning. I was looking to the binary images, but I guess it's not just that. There's more.
Yeah,
so I was trying to understand that.
So yeah, so I guess the way that I understand the cortical spectrum, the way that they define it, is just kind of like what we see in this image here. So the spectrum varies from regions that are where the cortical layers are not super defined to regions that are slightly more defined, and finally regions where they're super defined. So it's just the gradation like of structure, and then we can see the regions that have, for example, like V1L, that are super defined levels are kind of closely connected to V2 and V4, where I guess they're also quite strongly defined layers, but not as strong as V1. So I guess this is kind of what I understand by their cortical spectrum organization. Maybe I could be wrong, but yeah, I think it's just like a continuity of based on laminar features.
Okay. That it's kind of like similar, because to me, like I have in mind the Ulic maps, you know, the Ulic atlas, like also for the Broca area or the premotor cortex, when you see the different granulosity of it's something like that.
Yeah, I think so.
Okay.
Yeah, but I think maybe with the older atlases, like they always they mentioned even Brodmann earlier, where we have this like super strong differentiation between borders. And for me, when I kind of was really getting into neuroscience, that was a crazy shock that you can't really trust these Brodmann maps, where the numbers are quite arbitrary. And yeah, I think I'm definitely I'm really super excited about what they did. I don't think they have posted any. I couldn't find any code in their supplementary materials, but maybe I couldn't find it.
Just read MATLAB somewhere, but I was trying to look it.
Yeah, I think they just mainly use toolboxes on MATLAB, but I have to say modeling is not my strongest suit, but it looks super interesting. And I really love the fact that we can relate histology to something like MRI. That's a little bit more the bigger scale.
Yeah, it's really super interesting, the translational method.
I'm not surprised that MATLAB stands out to you, Leah.
It's very typical.
But I have to say, because that figure is up at the moment, I do love that they actually brought it back to the brain and actually showed the gradient on the brain, because a lot of papers use fancy modeling or fancy methods, and they combine things and they feed you that information, but never bring it back to a brain map level. So I think it's very nice to have this map as well. I was just, before I hand over to you, Valentina, I saw you. I have a question, and that is also about this map, because it's beautiful to see the map and see the gradient. But so what are we going to do with it? What can this map tell us that we don't already know, and what can we do with it to actually make use of it? Is that part of the paper as well? Do you have a guess?
Let me quickly have a look at my notes. I think, well, as far as I remember, they don't really make any bold claims like that. I think it's just more like, hey, this is a feature that we can extract from myelin staining for a computational method that doesn't have to be super laborious and spend hours on it. We can just shove it in an algorithm and it gives us this. But this sort of gradation we can relate to what we get from myelin maps from T1 and T2. So for example, strongest myelinated areas would be the sensory cortex and visual cortex, and then and then kind of less myelinated will be areas such as the association cortex. I guess that's kind of their main point, that you can see this cortical spectrum not only on the level of like laminar features, but you can also see it on the level of like myelin content that we can see with fMRI. I think that's kind of the main takeaway. But yeah, I don't really know. I have to say I'm really like I'm not super familiar with Sept architecture, but the one thing that they do note, because so this is only done on the macaque brain, there's still a possibility to do that on the human brain and maybe compare and have a look. There's also, we have many other primate species that we have such architectural data on that we can have a look at as well. So I think you could make a very interesting comparative study out of this.
Oh, that's for sure, yeah.
But yeah, that's kind of the main takeaway for me.
Okay, thank you. Valentina.
Yeah, my question was actually very similar. I was wondering how this reflects onto the cognition. So in which way we can use this information to wonder about so the organization of the cognition according to the various different laminarization of the cortex. But yeah, you answered me actually with the hypothesis of the myelinization. Yeah.
Sorry, didn't mean to steal your question. Oh, Evie, yes.
Yes. So thank you for the presentation. Just to add a question to that. So I was wondering if they talk about the next step that will be parcellation of the brain based on these T1, T2 maps. Given that, yeah, you can come back to the cytoarchitectonic map. So do they talk, because here, if I understood properly, they took different sites, precise sites. But if they go to a whole brain parcellation, did they talk if it is feasible to do this parcellation and to find the limitation between regions and stuff like that?
Yeah, so basically, essentially what they do here is they take the let me have a look at how many number of cortices they take. I think they take, well, I don't think they say how many slices of the whole brain that they take, but they take several. And the way that they define the slices is they're not right next to each other. They're based on the 78 regions that they're interested in. So they're not exactly next to each other. So they do say there's the continuity of this cortical gradient between the layers themselves is not continuous because you have this gap where they didn't take the slice. They don't really mention if you could just take the whole brain and like take slice after slice and have a look at that, but I think that would be super interesting to do. I imagine, like, obviously now when we have this, when they have this algorithm that is verified by also by specialists that have had a look at it by eye and have adjusted the thresholds, I think it wouldn't be, well, I guess it will be labor intensive as well, but I think it would make it a little bit easier to have a look at the whole brain in terms of this cortical gradient. But yeah, I don't think they don't really mention it as a next step post. That's what they will do. But yeah, they do mention that there's some continuity discrepancy in between the slices themselves. So they only look within slice, if that makes sense.
Okay.
Thank you, Evie. I was also wondering how that might, and there's probably no answer to this question just yet, but like gradients are a thing in neurosciences at the moment, right? So I'd be curious to see how this cortical spectrum maps or doesn't map onto the gradients as well, and try to bring it together at some point.
Yeah, maybe there
has
is
an idea.
parallelism. No, yeah, it's true. That it seems maybe
your face light up. Yeah, I think it would be interesting to see a comparison between it. There's definitely many studies that could come from this, like the comparative and the gradient and spectrum mapping and all of that. Great. Any more questions? No? Well, in that case, thank you all for joining us today. Sorry again for the tech issues earlier today. And I see you next week again for the next Neuroccino. I think Hervé is presenting next week. Yay, cool. Looking forward to it. Fantastic. Have a lovely and exciting week in science, and we'll see you next Monday. Bye.
Bye. See you.
Yeah, Leah, if you could stop the recording on my computer. Don't forget the live stream.